Hello, everybody. Thank you so much for your time for hearing my presentation. I would like to introduce myself. My name is Imran Roshadi from Department of Clinical Pathology in Gajah Mada University. Today, I would like to present my research about the clinical diagnosis and molecular detection of Anaplasma phagocytophilum for the first time in diary Ettawa code from Kalikasing, Central Java. So anaplasmosis in goats is one of the most serious problems, especially in the small ruminant sector. It can reducing the animal productions like milking or meats and also reducing the weight gain. So it can be a loss economy from for the farmers. Among the various anaplasma species, Anaplasma phagocytophilum are obligate intracellular bacteria parasitizing not only erythrocyte but also monocyte or granulocyte of higher vertebrates, mostly in ruminants. In the picture, we can see there are two types of bacteria that infected for the that infected neutrophil or polymorphonuclear cell. We call it Anaplasma phagocytophilum, and for monocyte we call it Ehrlichia capiensis. Both are very zoonotic, not only can infect the disease in animals, but also can infect diseases in the human. Especially in small ruminants, there are bee strain that maybe because of the closest life with the farmers, maybe it can be zoonotic soon. But the study about the Anaplasma phagocytophilum in small ruminants, especially in Indonesia, is very limited. In this research, I used 20 Ettawa breed coats which have a clinical signs, lethargy, weakness, reduced appetite, dull hair, pale mucus, heart rate, and respiration rate above the normal. From that animals, we samples the blood and also make a further analysis for the blood smear analysis, hemogram analysis, and molecular analysis to find the Anaplasma phagocytophilum that infected in dairy Ettawa breed coats. So surprisingly, around 40%, from 40%, we found intracytoplasmic forms or moruli inside the neutrophil in the dairy in the dairy coat at our dairy court. So it's look like similar the characteristic of of the Anaplasma phagocytophilum. And when we calculate the level of bacteremia, it's around 2.5 until 1%, which is belong to the subclinical. It is confirmed by hemogram analysis that all of the animals that have been positive from the anaplasma is still in the range normal of the hemogram from the reference. Also, but if we compare the anaplasma positive animals with the anaplasma negative animals, similar dairy codes in the same location, there are significantly differences in all of the parameters. Mostly the anaplasma positive code is have the lower values compared to the anaplasma negative animals. So it means that the anaplasma phagocytophilum in dairy cod cattle is still in the subclinical stage. And from the blood chemistry analysis, we found some interesting novelty that the anaplasma uh, can increase in ALP, bone, phosphorus, total protein, and globulin compared to the normal animals. So it means that the animals that have been infected by anaplasma have a liver damage. And we specify uh, it, the species that by using 60S rRNA, it's confirmed that this parasite or this bacteria is the anaplasma. But and then we more and then we need and then we do and then we did the nested PCR to specify the species it is belong to Anaplasma phagocytophilum or not, and all of the sample are positive to the Anaplasma phagocytophilum from the primer that have we been made to identify the Anaplasma phagocytophilum based on the cytochrome B. So in here can be seen there are 642 sites per pair. And for further analysis, phylogenetic analysis, we found that the parasites or the bacteria Anaplasma phagocytophilum that we found from the goat in Purworejo is a very similar, 95% similar to the Anaplasma phagocytophilum that have been found in sheep, cattle, and pig in China. So it is confirmed that this bacteria is Anaplasma phagocytophilum. So in the summary, egg Ettawa, around 40% have been infected by Anaplasma phagocytophilum. So, but still in subclinical stage, we have still the normal hemogram or normal value of the hemogram compared to the range normal. But if we compare to the negative animals, they have a significant differences. And from the chemical analysis, it showed that these parasites uh, make a liver damage. So in here we made a comprehensive study about the Anaplasma phagocytophilum that found in the small ruminants in Indonesia for the first time. I would like to thanks about this research to my Faculty of Veterinary Medicine, Gajah Mada University, for the funding, and all farmers, veterinarian, all staff in Department of Veterinary Clinical Pathology, Gajah Mada, and all staff in the Central Java Provincial Livestock and Animal Health Office that support my research. Thank you so much.